necessarily homologous landmarks. So instead, I just did two separate analyses, two-dimensional, and I delimited their face and brain case modules by development. So if you look at developmental series and kind of classification, you can figure out the modules, and then you can test the significance of the modules in Morpho-J or geomorph, whatever you want. So next. So after that, you need trees in order, you need phylogenies to study evolution. So we use I use a time calibrator tree from one of my colleagues, Victor Tagliacolio, and I used 134 species for this. For carnivorans, it was really sweet. They had this 76 gene super tree that was also time calibrated. So I just plugged all my skulls into that tree. And then I simulated evolvability, and I used the Landy equation. And instead of the additive genetic matrix, I swapped that out for a phenotypic variance-covariance matrix. And with that, I then randomly generated 1,000 selection vectors under a Gaussian distribution, and I multiplied the P matrix against beta, which are the selection vectors. From that, I generated 1,000 response vectors, and that gave me the ability to estimate evolvability, respondability, conditional evolvability, constraints, and autonomy. I could get into the definitions, but it's better to explain it to you in the results. So for rates of modular evolution, like I said, I split the skull down in half with face and brain case, and I measured them across 5 clades. But we won't really talk about that, but we'll see. I'll let you know. So as far as hypotheses go, if you're looking at a system that's integrated in evolutionary time, you should expect to see similar responses to selection vectors between modules, because the Plateau network will integrate any change that hits anything within that network. And as a result, you should see similar rates of evolution between the two modules, right? And if you're modular, you should expect more disparate responses to selection vectors, and you should expect more disparate rates of modular evolution. So let's see what we find. So the first result is integration. So this is the phylogenetic partial least squares analysis for genetiforms. And what you can see is genetiforms are actually pretty integrated in phylogeny. This is the correlation coefficient, 0.9. So they're actually pretty integrated. So we'll see what other implications that has on our later analyses. Within carnivorans, it's actually a very different pattern here. You can see that carnivorans are far more modular in their development, and there's almost, this is kind of like an outlier. If that was removed, this would go down even further. So yeah. So when we look at the evolvability simulations for genetiforms and integrated system, what you see is for the majority of these, you can focus on yellow here, but the majority of these are actually similar between the face and branchiates, which is exactly what you'd expect for an integrated system. However, the conditional evolvability and the autonomy, which are the ability to respond to selection in the presence of high integration, so that's a big deal, is much higher for the face, like an order of magnitude higher for the face. So the face has the ability to better respond to selection under the high integration than the brain case. So we'll see what that looks like when we get to rates of evolution. For carnivorans, what you see instead is that the face or the brain case actually exhibits way more evolvability, way more respondability, which is just the raw ability to respond and how readily you respond to selection vectors. So the brain case just outweighs that. Outweighs the face substantially. So when we look at their rates of evolution, all this kind of starts making more sense, because you can see that between the face and brain case and genetiforms, the face kind of just slightly edges out the brain case. Here, the ratio is like 1.2. And with carnivorans, the brain case evolved nearly twice as fast as the face. And why is that? Well, we'll get into that. So here's what we know so far. We know that the face of genetiforms is slightly faster than the brain case, and we know that the face can better respond to selection under the presence of high integration. And we know that the brain case in carnivorans has evolved twice as fast as the face. And we know that the brain case exhibited more evolvability and respondability. So why? So within genetiforms, I think this comes down to development. So within development, the underlying signaling structure that forms the face is usually really governed by the forebrain, and there are smaller factors that affect it, but the forebrain is really the big player here. The signaling from the forebrain of sonic hedgehog and FGFE, they really control the expansion of the facial primordia. So study by Hu et al. In 2003 found that if you perturb the signaling from the face of the brain case, which normally in development they get the signaling, the face expands. But if you remove that signaling, the face actually fails to expand. And you get left with these very brachyspheric phenotypes. And because the sonic hedgehog works as an integrating factor within the entire skull, it's controlled by a very strong quadruped network. So any hit to that quadruped network that cuts off the signaling or cuts off the reception of the signaling in the facial primordia would result in this foreshortened face. Therefore, this could possibly act as like an engine of variation that can generate these very disparate phenotypes. So what about carnivorans? So carnivorans exhibit rapid brain size evolution, like I mentioned before. And in development, the brain case closely tracks the brain, especially in mammals, where the brain fills the entire cranial bulb. And because the brain, again, plays such an important role in structure of the neurocranium and development, it's no surprise that the brain case is most likely tracking rapid brain size evolution in this way. So, let's synthesize. So, the genodiform skull is integrated in evolution. However, the face has the potential to generate variation by integration. Therefore, I hypothesize that the genodiform face slightly outpaced the brain case during evolution despite high integration because of this innate ability to kind of generate just a little bit more craniofacial variation. As far as the carnivoran skull, it's modular, and as a result of its modularity, the brain case is more vulnerable than the face, and due to its higher vulnerability and responsibility, it is able to evolve twice as fast, likely due to pressures tracking brain size evolution. With that, I'll take